Hello. Good morning, everybody. Is it working? Really? Alright. I cannot hear myself, but Melissa says it's working, so. Hello. Really? It's working? Alright. Alright. Is it working really? Alright. If Melissa says it's working. Whatever she says goes. Good morning, everybody, and thank you for coming. And also greetings to those who are listening in remotely. Before I introduce our 2016 awardee of the Wiese Distinguished Lecture Series, let me say a few words about this series. So this is made possible by a gift that the late Peter and Marty Wiese gave to the University of South Alabama and the C Lab. Peter and Marty were seriously committed to the environmental cause, and they were long-time benefactors of our program, and they were certainly highly esteemed by many of us. We miss them. So when Peter and Marty donated this gift, they made it clear that they wanted to mostly benefit the students, right? So they thought of you guys first. And the idea is to bring a preeminent scientist, well-renowned, to interact with you for a few days through talks and workshops and meetings. So that's really something that we should all take advantage of, because you're students. I'm sure that you're reading the papers and looking at your heroes and looking at people that you really value and work that inspires you, and it's a great opportunity for you to meet those heroes of yours and talk to them and learn a lot and get some inspiration. It's also good for all of us because it's truly a very unique learning and inspiring experience for all of us here. So, having said that, our 2016 Wiese speaker is Dr. Levin, Director of the Center for Marine Biodiversity and Conservation, also Distinguished Professor and Oliver Chair at the Scripps Institution of Oceanography, University of California at San Diego. Lisa's interests span a wide range of areas, from wetlands work all the way to deep sea ecosystems and ocean deoxygenation and acidification. As a matter of fact, Lisa's research has used, continues to use, large vessels, submersibles, ROVs to study these deep sea ecosystems. Dr. Levin has participated in over 35 oceanographic expeditions around the world, and she has been the chief scientist in half of them. Lisa's record is truly eminent. She has over 200 publications, many of them in leading journals. She has edited 5 special volumes on aspects of deep sea biodiversity. Her work has been cited more than 10,000 times. She has been on the editorial board of several leading journals. She has received ample international recognition, made evident by her participation in high-level steering committees, panels, and advisory groups. She is also a fellow of the American Geophysical Union and American Association for the Advancement of Science. Needless to say, her funding record is truly impressive, and she has graduated many students, one of whom we remember fondly, Joel Frody, who was a postdoc here and right now is an assistant professor at UNC. So Joel is one of your doing as well, Before I turn it to Lisa, I just wanted to add something that I think really speaks to the impact and relevance that her work has, and how applied it is, and how it can help managers develop effective policies for the use of natural resources, particularly deep sea ocean. And that's the Deep Ocean Stewardship Initiative that she recently funded. And what that initiative wants to do is to bring a lot of different people from different areas, including technology, policy, law, economics, to basically try to preserve the health of the deep sea ocean and make its use sustainable and environmentally friendly. So I think that is an example that us scientists should try to follow, make our work applicable, and her research is truly inspiring in that regard as well. Lisa, thank you for coming.
Okay, thank you very much, Wiese, and for that nice introduction. Can everybody hear me? Is the microphone working okay? Yes? It's really an honor to be here. And I'm just decreasing the volume. Go ahead. Okay. Yeah, it's a great pleasure to be here and to be able to talk to you today about another one of my very favorite subjects, which is oxygen. Those of you who know me know I've been interested in oxygen for many decades now. And I know that many of you here think about hypoxia. I tend to work in places that are very different from the Gulf, on upwelling margins around the world, so that's what you're going to be hearing about today. But I want to start with my acknowledgments, and the reason is that I tend to run a little long and rush through the end, but it's really important for me to point out that I've worked internationally and had many, many different colleagues that have enabled me to study oxygen and oxygen minimum zones, and they come from many countries. So I'm not going to read through this list, but I just wanted to point out that the work you're going to be hearing about today is not work that I did alone, but work that I've done with many different collaborators and also many students and technical support staff from my lab. And of course, I've been funded by different kinds of organizations as well. But let me start now talking about oxygen. Why should we care about oxygen? We know that oxygen has been the key to life on Earth for billions of years, and that there have been two really major oxygenation events that have had a huge influence on life. The first one happened over 2 billion years ago and led to the oxygenation of the ocean and the evolution of eukaryotes. The second event happened more recently, about 540 million years ago, and led to and was another oxygenation event. The first one was associated with the evolution of photosynthesis. The second one is still up for debate about exactly why it happened, but we do know that it led to this explosion of diversity among the eukaryotes. We call this the Precambrian explosion, and really it created all the major life forms that we know today. So we know that oxygen can be very, very important in stimulating the evolution of biological diversity. But we also know that the loss of oxygen can take away diversity. And in the past, we've had many ocean anoxic events. They're shown here in red through the geological record. And associated with these have been mass extinctions or massive declines in diversity over time. Probably the best analog, the PETM, occurred about 50-some million years ago, and is the best analog to climate change, the kinds of climate change that we are seeing today. But I really don't want to talk about the past in this presentation. I'm going to focus on the present and the future, and I'll first talk just some oxygen basics, the sources and distribution of low oxygen in the ocean, and then discuss what I've been able to learn over the past few decades from studying modern oxygen gradients on oxygen minimum zones in upwelling areas. And then the second part of this presentation, I'd like to talk about changes to oxygen in the ocean, a phenomenon we call ocean deoxygenation, and then I'll talk a little bit about this in a multi-stressor context, and then close with some thoughts about what this means for managing our continental margins. So I'm going to start with some basic information that I'm pretty sure most of you in this audience know. Where does our oxygen in the ocean come from? Well, a lot of it comes from the air. It's mixed into the surface waters at the surface of the ocean. And other important source of oxygen is photosynthesis, which is carried out by phytoplankton in the surface ocean. Now, oxygen enters the interior of the ocean through thermohaline circulation. This happens over hundreds to thousands of years. Water sinks at high latitudes, both in the North Atlantic and at the poles, and then moves in the interior waters towards the equator. So this brings oxygenated water down from the surface into the ocean, interior of the ocean. I want to say just a few words about measuring oxygen. There are as many kinds of oxygen units as there are kinds of scientists that study oxygen. Excuse me, I'm going to need to take a drink here. So, many of the biologists use milligrams per liter. How many of you think in milligrams per liter? Raise your hands. How many of you think in milliliters per liter? Oh, nobody. How about micromolar? A few. Okay, so those are concentration units. I'm just seeing that the micromolar didn't come out in the PC to Mac conversion. I didn't catch that earlier, but the chemist should say micromolar. And then so those are different concentration units. The physiologists tend to think about the influence of temperature and pressure on the oxygenation experienced by the organism. So we have PO2 and percent saturation in kilopascals. And every study is done in different oxygen units, and there are conversions. And I'm going to try to give the conversions when I think it's important. But the second thing I want to mention here in terms of terminology is what is hypoxia. We think of this as a shortage of oxygen for supporting life. And most people who work in coastal waters have a definition of hypoxia that's associated with 2 milligrams per liter, 1.42 mils per liter, or maybe 63 micromolar. And this has evolved, I should say, I believe it's a fairly Atlantic-centric shallow water definition. Those of us who work in naturally occurring low oxygen areas in deep water tend to have a cutoff of around half a mil per liter, 0.7 milligrams per liter, or 22 micromolar. This is the definition of oxygen minimum zones. This tends to be Indo-Pacific centric because that's where you find the majority of those kinds of settings. There are some in the Atlantic. So these are just two thresholds to think about. They don't apply to all organisms. Now, how do we get hypoxia in the ocean? It turns out we get it the same way in the coastal and estuarine ocean as we do in the open ocean, and it usually starts with the input of nutrients, often nitrate, sometimes phosphate. In the open ocean, this comes from upwelling of deep nutrient-rich waters into the surface. In coastal oceans, it can come from upwelling, but it also can come from inputs from land, from runoff and fertilizers, for example. Once we get the inputs of nutrients, this usually stimulates phytoplankton production, and sometimes this leads to phytoplankton blooms. And as the phytoplankton are either consumed or die and decay and sink, respiration takes place, mostly microbial, but not completely. And of course, we know that respiration consumes oxygen. It also releases carbon dioxide. This intense oxygen consumption doesn't necessarily lead to hypoxia if there's a source of oxygenation, if there's oxygenated waters coming into the system, if there's mixing or maybe currents coming through. However, when those are absent, we tend to get the oxygen being used up, and we end up with hypoxia. Now, this takes place in estuarine and coastal waters as a result of eutrophication, and I think people here know this very well. Bob Diaz has been tracking this for a long time. In the 1960s, the northern hemisphere showed quite a few hypoxic areas. 20 years later, there were even more hypoxic areas, and 20 years after that, even more. You can see, again, very intense hypoxia along the margins of the northern hemisphere, not so much in the southern hemisphere. If we look at naturally occurring hypoxia that results from upwelling on margins, we find that there are these areas that you see here in pink that where we see low oxygen between about 100 and 1,000 meters. These are regions not only of upwelling, but they're areas where the water tends to be old. It's been isolated from the surface for a long period of time, and there is no source of ventilation or major oxygenated currents coming in. And what's really interesting is if you overlay the natural hypoxia in the ocean, where it intercepts our continental margins, and that's the area in black, with the eutrophication-induced hypoxia, what we find is that they often don't overlap that much. Some, like the U.S. West Coast, they do, but that much of the ocean's continental margins actually experience hypoxia of one sort or another. So it's kind of not everywhere, but it's a very widespread phenomenon. So as I mentioned, where oxygen minimum zones intercept the margins, we have hypoxia. We develop hypoxic seafloor. This is represented here in this yellow ribbon along the coast of Mexico, the west coast of Mexico. And I've worked with John Helly to calculate that globally, very, very conservatively, there's over 1.1 million square kilometers of continental margin that are naturally hypoxic with oxygen levels less than about 0.7 milligrams per liter or 0.5 mils per liter. But where this occurs, the water depths at which this occurs really vary. And here's an example of the distribution of the oxygen minimum zone in the eastern Pacific. And you can see that it's actually a very deep phenomenon off Canada, Washington, and Oregon, and California. It doesn't really start till 500 or so meters. And that's because we have the California current and other oxygenated currents bringing in oxygen up here. Off Mexico, it's a very thick feature, but around the equator, we have an oxygenated equatorial current that deepens in it again, and then it thickens again off Peru, and it eventually peters out off central Chile. But this phenomenon then occurs at different depths, at different latitudes. Now, I want to talk just very briefly about my own interest, how I developed an interest in naturally occurring low oxygen. And it really, truly happened by accident, just serendipity. I was working on seamounts in the 1980s and made a dive with Alvin on a seamount called Volcano 7, 200 kilometers west of Acapulco. And we were studying the effects of seamount current intensification on life. And I was in Alvin, the 3-man submersible. We came down at 900 meters, and it was a very lush area. The sediments were green with phytodetritus. There were a lot of fish, a lot of shrimp, really high densities of animals. And we started climbing the slope of the seamount towards the summit. And it was very clear that there was sharp zonation. We left this zone of fish and shrimp, and we got to a densely packed brittle star zone. These are really old photographs, transferred from slides, so they're not so great, but you get the idea. Then we left those, came up 50 meters, and found a zone covered with sponges and crabs and serpulid worms, completely different assemblage. So there was really sharp zonation. Then we came up another 50 meters, and there was nothing. The seamount was this was the top of the seamount, and it was really, there was some fuzz on the rocks, and occasionally a fish came through. You can see fish in this picture, but there were no animals. And so I was very perplexed about what caused this sharp zonation and this absence of fauna. And at the time, I totally did not have oxygen on my radar screen. But it turns out this volcano was protruding up into the oxygen minimum zone. And somewhere between 800 and 750 meters, we crossed the oxygen threshold. The oxygen was getting lower and lower as we went up into that zone because it's better oxygenated beneath the zone. So we crossed that threshold, and we totally lost the megafaunal and macrofaunal life forms. And I figured this out not long after the cruise, but at the time, I hadn't been out there to study oxygen. So this was just a really interesting finding. But it set me off really on a track for the next 25 years of trying to look at the effects of naturally occurring low oxygen on ecosystems. And here you on naturally continental margin ecosystems. And you can see the many areas that I've worked, and I'm going to be talking today about some of that accumulated knowledge where I've had the opportunity to study cross-margin transects that run through oxygen minimum zones. A really important point is that these are not dead zones. There is, for the most part, there is life there. And I get kind of upset when people call these dead zones. They are naturally occurring low oxygen areas. But the animals there have experienced persistent hypoxia and evolved amazing adaptations to living at low oxygen. So we find many, many species with red blood and hemoglobin, even taxa like sea cucumbers and mussels, things that normally don't have hemoglobin, we find with high oxygen affinity hemoglobin. We also find many animals with enhanced surface area, like this terabellid polychaete with the branchial structure that's almost as big as the worm itself. I like this one from off Costa Rica. And there are many animals with expanded surface area to volume ratios. We see this in the form of branchiae and frilly gills in the amphipods, and even the nematodes are frilly out in oxygen minimum zones. So the fish have frilly gills. There's all sorts of ways they have evolved morphologically to enhance oxygen uptake. Having long, thin bodies shapes also increase the surface area to volume ratio for oxygen diffusion. So there are many adaptations among these organisms. Now, my first studies were conducted on Volcano 7, and what I learned from this very early work was that oxygen gradients can cause body size zonation. Turns out at that surface of the volcano that I showed you, where we saw not much animal life, the microbes and the myofauna actually thrived. There were tons and tons of animals. These are from sediment cores. It was the macrofauna, animals retained on a 0.3 millimeter mesh, and the megafauna, animals that we can see with our naked eyes, that were actually depressed on the top of the volcano. However, just 50 meters below, they also had very high densities. And you can see from the oxygen gradient, sorry, guys who think in milligrams, they're not up there, but you get the idea that there's really not a huge oxygen change from the top to the lower summit, but that there is a threshold there that enables the animals to occur. So this density maximum, it turns out that I saw in Volcano 7, is a widespread phenomenon. You can find it in oxygen minimum zones all around the world. And so I've plotted here macrofaunal densities across oxygen gradients. The OMZs are the gray shaded area. And you can see that in many, many cases, almost all cases here, we have peak densities somewhere within or near the lower boundary of the oxygen minimum zone. And this occurs where oxygen crosses a threshold somewhere between 4 and 9 micromolar, where it just pops up, so 0.1 to 0.2 mils per liter, and that enables a really high density of animals that are just able to tolerate that system and have the advantage of lots of food, but no predators in many cases. We also have found across oxygen minimum zones that there are really, really sharp diversity gradients. These are rarefaction curves that show the number of species expected as a function of sample size. And in all cases, we have really low diversity in the core of the oxygen minimum zone. And as you move, usually across the lower boundary, oxygen increases slightly and diversity increases dramatically. And the interesting thing is this was first shown in Walvis Bay by Howard Sanders in a 1969 paper where he sampled across oxygen gradients on the Namibian margin. Not many people paid attention to that, but this type of pattern has been repeated over and over again since. So I've been showing you some of the structural attributes of communities that live across oxygen gradients. But my opportunity to really understand how that structure translates into ecosystem function came when I collaborated with the British to work on the Pakistan margin, which is not an upwelling area, a traditional eastern boundary upwelling zone, but it has monsoonal winds that create upwelling seasonally. And so it does have a very well-developed oxygen minimum zone. And what we see off Pakistan, this is the oxygen profile. It's very well oxygenated at the surface. It drops very sharply between 1 and 200 meters. And then there's a core region of extremely low oxygen, and then it starts up very, very gradually at about 700 or 800 meters and increases again in deeper water. And I want to focus on these two areas, and I'm going to start with this transition zone between 700 and 1100 meters. The upper area is important because seasonally the oxygen levels shift from being well oxygenated on the shelf to being very poorly oxygenated after the monsoon. Okay, so if we look at just at the abundance of different sized taxa across the Pakistan margin, what we find is that the foraminifera, these are single-celled protozoans. That's the definition. These are protozoans, single-celled animals. They dominate in this really low oxygen core, but they too have a peak somewhere at around 700 meters, where oxygen just begins to rise ever so slightly. The metazoan macrofauna are absent in that core region, and they kick in at about 800 to 850 meters. The mobile megafauna invertebrates and the benthic species basically kick in a little bit deeper at about 900 meters or 1,000 meters, and the nekton show up and peak at about 1,000 to 1,100 meters. So there's a clear body size zonation that happens across these oxygen gradients. But what's so interesting is this oxygen threshold, the changes are only 5 micromolar between 700 and 1,100 meters, or 0.1 mil per liter. I'm not sure what that is in milligrams per liter. It's probably like 0.15 or something along those lines. So it's a really small threshold. We see among the macrofauna that there's a really strong diversity gradient from one species at 800 meters to many species at 1,000 or 1,100 meters. Again, just across this very narrow threshold. And these changes are associated with a sharp change in bioturbation. The sediments are fully laminated. These are X-radiographs of the sediment cores, by the way. They're fully laminated at 700 meters. And you can see they're laminated all the way through to about 850 meters. And then we start to pick up quite a few animal burrows. And by the time we're at 1,100 meters, the sediments are fully bioturbated. Again, just across a very narrow threshold. A small change, 5 micromolars, from 700 to 1,100 meters. And I want to just home in. It's kind of interesting. Some of you are interested in animal-sediment interactions. This area, you can see some burrows, but the sediments remain laminated. And this is where we saw very high densities of amphinomid polychaetes in the genus Linophorus. And we were really interested in how can you have high densities of worms and still have laminated sediments. And this is a picture from inside the lander at that depth. And the main thing I want you to see is that it's not a very clear, but the worms are making burrows, but they're sitting half in and half out of the sediment, and they're not really bioturbating or eating their way through the sediment. We've done experiments. They're collecting phytodetritus at the sediment-water interface, and they're waving. You can see them, waving back and forth there in the flow because the lander stirs the water. But in any event, it looks like you can have laminations retained even as you have some burrows present. So one of the questions we were interested in is how does oxygen and how does carbon processing change across oxygen gradients? And to ask that question, we use C13 labeled phytodetritus introduced into sediment cores on board ship. We collected these cores from different water depths, from different oxygen concentrations, and introduced the phytodetritus, and then looked at uptake by the protozoans, the foraminifera, and by the macrofauna that were in the cores. These were short-term, just a few-day experiments. And what we found is that oxygen has a huge effect on who gets the carbon when phytodetritus falls down to the seafloor. And so there's probably two things to point out here. The first is that we did this experiment pre-monsoon and post-monsoon with 140-meter cores. After the monsoon, the oxygen levels fall tremendously. And what we found is that before the monsoon, that it was mainly macrofauna that took up the phytodetritus. After the monsoon, it was mainly foraminifera. And that's the after monsoon results shown here. You can see foraminifera dominating the carbon uptake. The other thing we found is that foraminifera always dominate at 300 meters, where oxygen is exceedingly low and never changes. But that when we got to 940 meters, where the oxygen started up again, the macrofauna were the dominant carbon processors. So we see along a spatial gradient and in a temporal change that there can be a shift between protozoans and metazoans in terms of who processes carbon. I also had the opportunity to look at the effect of oxygen on colonization of benthic macrofauna. We did some short-term experiments on the Indian margin. These were only 4 to 8 day experiments. They were intended to be longer, but we had some problems with the Indian government. I collaborated with the Japanese, and we used the Shinkai 6500 to do these experiments. We put out colonization trays across an oxygen gradient, and basically the short summary of what we found is that where oxygen is exceedingly low, 0.1 micromolar, we got no macrofauna and no macrofaunal colonization. An order of magnitude higher oxygen, 1 to 2 micromolar, we got almost no colonization compared to the background fauna. But that at 1,100 meters, where oxygen is, again, an order of magnitude higher, 20 micromolar, we got, not only did we get a lot of colonization, we got an overshoot of the background in 4 to 8 days. So oxygen seems to have at least some control on the colonization potential of the fauna in these deep environments. I'd like to talk a little bit about the influence of oxygen on trophic ecology. I know many people in the audience here study trophic interactions of species, and these are 3 topics I want to talk about: the evolution of carnivory, trophic diversity, and chemosynthesis. I've worked with Eric Sperling, who is sort of half paleo, half modern ecologist interested in oxygen, and work with him has shown that the Cambrian explosion that I was talking about at 540 million years is associated with the advent of carnivory. And one explanation is that because carnivory is a very oxygen-intensive process, that the introduction of more oxygen into the atmosphere enabled the diversity of life forms that including carnivores. And the very first evidence of carnivory really shows up at about this time, and we see it in the form of boreholes, carnivorous species like Nemertians, and chaetognaths. So this is at least one hypothesis that that oxygen actually limited the evolution of carnivory and that it enabled it to occur. And Eric actually sought me out and teamed up because we wanted to ask the question whether we see this same pattern in the modern. In other words, do low oxygen areas lack carnivores? And so we collected data, cross-margin data from oxygen minimum zones for 68 stations and 1,100 plus polychaete species, and asked this question. And sure enough, what we found is at the very, very lowest oxygen levels, again, sorry, there's no milligrams here, it's 0 to 0.2 mils or 0 to 9 micromolar, we see that, in fact, there is a significant decrease in the number of carnivorous taxa and a significant decrease in the percent of carnivorous individuals. And in fact, half of the stations at these really low oxygen levels had 0 carnivorous taxa present. Another interesting question is whether this low species diversity and the few number of carnivores translates into low trophic diversity. Are there fewer trophic modes and fewer trophic interactions? And I approached this by using stable isotopes and using community metrics. So I I was able to collect stable isotope data on many of those cross-margin cruises. At the time, I wasn't too sure what I was going to do with the data, but I've just begun this analysis, and it's really sort of preliminary. But what we're looking at is carbon range as an indicator of food sources, and nitrogen range as an indicator of trophic levels, and also the distance to the centroid, where I should point out each of the species on the plots on my plots for which I did this analysis were average isotope signatures of each species at a particular station. So we can look at distance to centroid to represent species packing, and we can look at the spread of species on a isotope plot within a station and calculate the standard elliptical area to look at another measure of trophic diversity. So we had four basic stable isotope metrics of community trophic diversity, and I then sorted them by oxygen level across, developed from the stations you see, or the cross-margin transects you see on this map. We had 29 stations from 7 different margin areas. And the question was, does trophic diversity go down in low oxygen areas? And the answer is no. In fact, it might be higher, because I've not really done rigorous statistical testing at this point, but in general, the low oxygen areas have higher metrics for all of these, indicating that, in fact, trophic diversity is probably higher than it is in better oxygenated areas. So the answer is, at least based on stable isotopes. So why might that be? I think one of the answers lies with the occurrence of chemosynthesis in naturally occurring low oxygen areas. In many of these areas, there are chemosynthetic sulfide oxidizing bacteria that form mats over the bottom. Here's an example from Namibia. Here's an example from the Chile margin. We know these mats are important in detoxifying sulfide, and they enable the presence of a number of different kinds of polychaetes to be present, and they support a pretty important food chain that starts with bearded gobies and in Namibia and goes on to hake and horse mackerel. I talked about that a bit last night. But we do know there are chemosynthetic microbes. If we look at stable isotope signatures across some well-developed oxygen minimum zones, for example, northern Chile, we find that all the species collected above and below the oxygen minimum zone have heavy carbon and nitrogen signatures indicative of a photosynthesis and phytoplankton-based food chain, but the animals collected and even the bacteria collected from within the oxygen minimum zone, which is really just from 200 to 300 meters off northern Chile, then they all have very light signatures indicative of chemosynthetic food sources. And these are the kinds of animals. Some of the animals have symbiotic bacteria. We find them in this oligochaete olavius. In many of the bivalves, thiacerid and leucinid bivalves reach very high density sometimes in oxygen minimum zones. The mussel acarax, the bivalve acarax, and then what frenulates, we used to call them pogonopherans, are also really common. And they all have symbiotic chemosynthetic bacteria, and they contribute to that broader stable isotope signature in OMZs. Olavius is probably my favorite of these animals. I don't know how many of you, maybe if you've seen me talk, you've seen this slide before, but this is an example of an oligochaete that's lost its mouth, gut, and anus, doesn't feed at all, but inside, beneath its cuticle, are 6 different bacterial types that carry out sulfide oxidation, sulfate reduction, and spirochetes that are probably fixing nitrogen, and more work by the Germans has shown that basically these microbes carry out all the life processes for this worm, and the worm just migrates up and down and exposes it to the right levels of sulfide and oxygen, and the bacteria take care of its waste, fix carbon and feed it. It's like a whole ecosystem inside this one oligochaete, which is really, really common in off Peru and Chile in the oxygen minimum zone. So these are examples of structural and functional control of oxygen on margins that we can learn about from naturally occurring systems. But we've had a very static look at this system. But now I want to talk about oxygen in a changing ocean, we know that oxygen is not static, that it's changing over time, and I'll talk about these different aspects of this. And I think that, you know, I have to say that I was working in these systems for several decades before anybody really became interested in them. It was sort of basic esoteric science at some level, but that all really changed in 2008. There were a couple of very important papers that came out that showed that naturally occurring oxygen minimum zones were expanding, and that especially off California, where we have really good records, that we've lost significant amounts of oxygen. At 200 to 300 meters, we've lost 20 to 30% of our oxygen in the last 25 years. So big effects. So people started paying attention to oxygen as a form of climate change. There was a really nice summary paper that came out in 2010 by Ralph Keeling and co-authors that defined the term ocean deoxygenation, and it explained how this was happening. We began to see climate negotiations, talks on one of my students gave the first talk, in fact, of ocean deoxygenation in Warsaw, and we had more talks again at COP20 and COP21. There were papers that started to come out that talked about the human impacts of oxygen loss in the global ocean, this one by Moore et al. And others. And then there's been a whole series now of more recent papers that have addressed both the changes in oxygen globally and the consequences for biological systems in the ocean. So I want to talk about this a little bit. But I do want to point out that this loss of oxygen is not just an oxygen minimum zone expansion. It's occurring pretty much everywhere, and it's not only because of eutrophication, although some of it is. Where we have really long-term oxygen records, and here I show examples, you can see steady declines in oxygen in the Adriatic. There are long-term records from the Cariaco Basin, from the bottom of San Francisco Bay, from the bottom of the East China Sea. We see massive increases in hypoxia in Chesapeake Bay, in the Baltic. In the Gulf, you can see it's a lot more variable, but certainly it looks like there's increases since this time period. So coastal and estuarine areas have been experiencing ocean deoxygenation, some of it from, and I don't have time to go into it in this presentation, much of it's from eutrophication, but some of it is from the effects of warming and the follow-on effects of warming, and I'll talk about these. We also know that oxygen has declined tremendously in the Northeast Pacific. There's a really good time series in the open ocean at Station P that shows since the 1950s actually, a lot of variability, but a loss of oxygen averaging about 2/3 of a micromole per year over time, and this continues. And then we have long-term records from British Columbia and Oregon and the Cal Coffee time series off Southern California, and they all show oxygen declines along the margins of the Northeast Pacific. Strama et al., I mentioned this 2008 paper, showed for 6 different areas in the global ocean that oxygen minimum zones are expanding. These are the areas in blue here. And this really got people thinking hard about the global phenomenon of oxygen loss. Strama and co-workers, and I was one of these authors on this later paper that took a look more synoptically at the ocean, and it's very clear that there's an increase in apparent oxygen utilization. Let me go back, see if I can do that. Oh, come back. Okay, there's an increase. These areas shown in red correspond to losses of dissolved oxygen, these areas in blue, through this broad swath of tropical and subtropical waters, and you can see also along much of the eastern Pacific. And this has been most intense at intermediate water depths, 200 to 700 meters, where oxygen consumption is intense, and I'm going to talk about why this is happening. But it's been estimated that, for example, at 200 meters, the area that's hypoxic or nearly hypoxic has increased by 4.5 million square kilometers since the 1960s. So this is massive global oxygen loss. We also have Earth system models that are projecting changes in oxygen to 2100. And you can see that these are under two different emission scenarios. Under business as usual, there's very intense oxygen loss in the North Pacific and much of the Southern Ocean and parts of the North Atlantic. In contrast, it's not quite so bad, but there's still oxygen loss predicted under the very best emission scenarios. So with global warming, we're on a track to loss of oxygen. And we know this is quite likely when the IPCC includes in its last report the Intergovernmental Panel on Climate Change, for those of you who don't know the lingo, that is really a global consensus of scientists that the expansion of hypoxic regions, termed oxygen minimum zones, and anoxic dead zones, referring to the coastal eutrophication observed over the last 50 years and projected into the future, is going to increase. And that's with medium confidence, which for that group is a lot of confidence, believe me. Okay, so here's a simulation, or not. It's actually not a simulation. I think it's real data of carbon dioxide emissions in 2006 to 7, and you can see most of it comes from the northern hemisphere. And so the question is, how do we get from rising carbon dioxide in the northern hemisphere to global oxygen loss throughout the ocean? I want to address this just briefly for a minute. It really comes down to issues of solubility, ventilation, respiration, changes in upwelling and nutrient input, and changes in circulation. So there's a lot of factors at work with rising CO2 and warming oceans. We know the ocean is warming, and I don't have time to show you all the evidence of that, but it's warming significantly. And we know that warmer seawater holds less oxygen. And you can see, just looking at the saturation levels, that at 100% saturation occurs at much lower oxygen levels. So 100% saturated water is going to have a much lower level in milligrams per liter than in warm water than it would in cold water. The other important factor is that warming raises the oxygen thresholds for living organisms. So the tolerance to low oxygen diminishes as the water gets warmer, and this shows the 90% thresholds for animals, for known sensitivities of animal species. So warmer water holds less oxygen, but the sensitivity to that oxygen level is going to be increasing among species. The other thing that happens in a warmer ocean is that it's more stratified. So there's intensified thermal stratification, and also melting of ice caps and glaciers leads to surface freshening, and that also intensifies stratification. And that means that there's less ventilation of oxygen from the surface waters into the interior. And so there's less mixing of oxygen in the ocean. And that actually, I should say, the warming and solubility effect is thought to account for 15% of the global oxygen loss in the open ocean, and this ventilation effect is thought to account for 85% of the global oxygen loss in the open ocean. But there are other things going on as well. As the planet warms, the temperature differential between land and sea increases, and this is intensifying upwelling winds. And so in the upwelling, eastern boundary upwelling areas of the world ocean, the 4 areas you see here, the majority of them have experienced intensified winds since the 1990s and increased upwelling of low oxygen waters onto the shelf and onto surface areas. So that's the retrospective look, but then now there's modeling of those upwelling areas, and those model projections to 2100 show that 3 of the 4 upwelling areas are going to only experience greater intensity, more upwelling, and more hypoxia. And the only area they're not so sure about is the California current, actually. But it's not all happening at the surface. I should say, and those of you who heard me talk last night, I mentioned that ocean warming is actually going to decrease, ocean warming is going to increase the dissociation of buried gas hydrates along continental margins, releasing more methane. And as that happens, aerobic methanotrophic bacteria are going to be consuming that methane in the water column, and that also is another source of oxygen drawdown in deeper waters. This often happens on margins between about 500 and 1,000, 400 and 1,000 meters. So that's another source of oxygen loss in the global ocean that's likely to affect all these areas on margins where we know that there are buried gas hydrates, including in the Gulf of Mexico. So what are the consequences of all of this oxygen loss, of expansion and shoaling of oxygen minimum zones and increased hypoxia for benthos? Well, we know that the least tolerant taxa are fish and crustaceans, and that hypoxia can induce mass mortality when the fish and crustaceans don't have anywhere to go. And I guess my understanding is there's something here called the Jubilee that probably reflects animals moving toward shore or flopping out onto the shore when oxygen gets low. I'm not 100% sure that's the cause. Is that the cause? Yeah. But we see these kinds of effects around the world. So in the Benguela Current in South Africa, the lobsters walk out onto the shore and, they're a foot deep, basically, when they have algal blooms and hypoxic events. On the Oregon off Oregon now, we have seasonal hypoxia that never really used to happen, and in 2006 there was major mass mortality. This is a picture from Southern California where we had a massive sardine push into the harbors and a massive kill. So there are these big mass mortalities associated with hypoxia. But there's a lot of other things happening that are much more subtle. And Deutsch et al. Had a recent paper that looked at changes in the ocean's metabolic index. They define this as the ratio of oxygen supply to resting metabolic oxygen demand, and show that this index is going to be increasing with warming. We know that I mentioned that the sensitivity of species to hypoxia increases. In other words, they're more and more vulnerable at a particular oxygen level, and this is shown out here for all these different kinds of taxa as a function of temperature. But what Deutsch et al. Modeled was what would change to fish survival and mortality with warming ocean. And they did a 1 degree and a 4 degree scenario for warming, found reduced fish survival times under hypoxia would increase 10 to 36%. Again, pulling hypoxia is that probably 2 milligram per liter value. And that they would be increased thresholds for oxygen concentrations that would induce high fish mortality. They predict that the fish in the ocean will exhibit both poleward and vertical contraction, and that this is really going to have a huge effect, potentially a huge effect on not just the ocean's fisheries species, the commercial fisheries, but probably all the fish species in the ocean. And one of the groups that's going to be particularly impacted in the open ocean are the diurnal vertical migrating fish and invertebrates. It's very interesting. It seems that seawater oxygen concentration is the best single predictor of their vertical migration depth at the global scale. Bianchi et al. Produced this map. And if you look at it, it really looks like a map of the world's oxygen minimum zones, but it's a map that shows how deep the vertical migrators go. And what is going to happen as oxygen minimum zones expand and world oxygen levels decline, these species are going to be migrating to shallower and shallower depths each day. They go down at night. I think my next slide shows us that these taxa that migrate, they spend their daytime either in the oxygen minimum zone or at the upper boundary of the oxygen minimum zone, and they migrate back up at night to repay their oxygen debt. But they're going to have to migrate up into shallower waters as OMZ expands. Another problem is that in surface waters, they're going to be exposed to warmer surface waters and higher CO2 levels from ocean acidification, so that there's a sort of a squeezing, a habitat compression from both ends, and it's likely that this is going to change species interactions and lead potentially to higher mortality rates. And Tony Kozlow has been studying this in mesopelagic fish and found very dramatic declines over the last 25 to 30 years in the abundances of mesopelagic fish. He first introduced this in 2011. There was a 2015 paper that came out that showed this community has declined by 70% as hypoxia is expanded. And he thinks it's due to increased predation in surface waters due to habitat compression. It's a big effect because these fish are essentially the breadbasket of the ocean. You know, they're feeding all the other fish that we depend on, the tuna and many other species that we harvest. But it's not just the pelagic system that's experienced habitat compression due to hypoxia. There's work by Whitney and Sinclair that's predicted that the groundfish species that we harvest commercially are going to lose half of their habitat area as hypoxia expands. By 2050, this will occur off California, British Columbia, and Alaska. And it's already occurred off Oregon, where there's been significant oxygen loss and shoaling. And then McClatchy and co-workers have also looked at cowcod, which was a commercial fishery, but they've closed it because this species loses something like 30% of its habitat every summer. We have seasonal hypoxia expansion off Southern California on these, and it's predicted they're going to continue to lose more habitat by the middle of the century. So this is a concern for groundfish species. It's not just the bottom fish being affected. We have evidence the benthic invertebrates are affected by the expansion of hypoxia as well. So some species that are intolerant are experiencing habitat compression, like this shallow or dwelling Lidochinus pictus, which over time, I should mention that there have been, Southern California Coastal Water Research Project has done trawl surveys every 5 years in Southern California. My student Kirk Sato has been examining the data, shown that several species have had a shoaling depths over that time period, over the last 20 years or so. Another burrowing urchin, Spatangus californicus, also shows shoaling depth, and all of these species have declining densities as this is happening. So they're experiencing a habitat compression. But we do have a very climate change tolerant species, Strongylus entrotus fragilis, that in fact hasn't been changing its depth and it has been increasing its density. And seems to live very well under these low oxygen, high CO2 conditions that it experiences. So this is just one example. Most people haven't really been studying what benthic species are doing in these open ocean margins over time, but this is an indication that there are changes. We also know that hypoxia is affecting the species that use the coastal or estuarine areas as nursery grounds on the West Coast. There was a nice paper by Wiese et al. that compared hypoxic and normoxic periods for fish species that use the Elkhorn Slough off Central California. You can see recruitment is much lower during hypoxic periods than during normoxic periods. These fish recruit offshore but use the estuary as nursery grounds. Also, species richness is lower during hypoxic periods. And so it's not just the fish populations and communities that are affected, but even individual ambits are affected by hypoxia. Rankin et al. Had tagged rockfish and shown and measured oxygen at the same time and shown that their foraging area is severely reduced. Each of these is a different tagged fish here. For the most part, their foraging area is reduced under hypoxic conditions than it is under normoxic conditions. And I could go on and on about the many different kinds of impacts of oxygen, but I want to take the last few minutes of this presentation, I'm running kind of long here, to point out that oxygen doesn't really act alone, that it's occurring in a multi-stressor ocean. We know that there's warming happening at the same time, ocean acidification, changes in food supply, especially predicted for the deep ocean. And if we look at model projections of changes of these major climate variables on the globe, you can see that many areas, especially the North and East Pacific, the North Atlantic, and the Southern Ocean, are going to experience multiple stressors, not just oxygen loss or not just warming, not just rise in declines in pH. And so one question is, with all of this happening in the field, how can we untangle these interacting climate variables, and what's the effect of oxygen? What's the effect of these other variables going on? And I want to just mention a couple approaches to doing this, and I want to show you some work we've done using natural gradients in modern settings, but also mention that people are starting to do complex laboratory experiments and apply other techniques. Now, this paper just came out about multiple stressors, and it's a meta-analysis of the literature. And what I really want you to look at is that other than the interaction of hypoxia with temperature, there's really very little literature out there about how low oxygen interacts with other stressors, and almost nothing. There's nothing shown, less than 15 studies on interaction with ocean acidification, as an example. But there's a lot of natural variability in the ocean that could allow us to ask questions about this. And oxygen minimum zones in particular, shown here, are actually carbon maximum zones. The same process, respiration, that uses up the oxygen, releases CO2. So we have huge carbon dioxide gradients along the same margins that we have low oxygen gradients. And if you look at the map, you can see that DIC map you have high CO2 in the same places that we have low oxygen. And the other thing is that our world's oxygen minimum zones span a huge temperature range. We have warm ones, we have cold ones, we have intermediate ones, and so we can use this natural variability in climate variables to ask questions about the effects. And I've done this with Eric Sperling and Christina Frieder using, again, our OMZ margin stations. We had 95 stations in which we were able to look at diversity data for macrobenthos. We looked at H' and J' and used variance partitioning tools to ask the question. We collected oxygen, PCO2, and temperature data for these stations and asked the question, can any of these climate variables explain diversity patterns? And these are the kinds of biota we're looking at, infaunal polychaetes, bivalves, ophiuroids, crustaceans, all the usual suspects, but on open ocean margins. And I'm just going to go quickly to describe the major results. And what we found is that in the East Pacific, and for the full data set, oxygen was by far, these are the result of random forest analysis, and each of these show the relative contribution of these variables to variation in H'. And basically, oxygen is the most important in the East Pacific, and these partial dependence plots show that the oxygen thresholds occur at really low oxygen levels, between around 7 micromolar, 0.16 mils per liter, maybe 0.2 milligrams per liter. So diversity is very low below those levels, and it shoots way up. But interestingly enough, in the Indian Ocean, oxygen was not the primary explanatory variable. I should point out that the oxygen threshold, it was the second. And the oxygen thresholds in the Indian Ocean are actually higher. They occur at about 0.5 mils or 0.7 milligrams per liter. And instead, PCO2 is the biggest explanatory variable with strong thresholds at 900 microatmospheres. So different ocean basins, possibly different controlling climate variables on benthic biodiversity. However, when we looked at evenness, we found that temperature was by far the dominant climate factor that seemed to at least explain more of the variation, although total variability explained wasn't very high in these studies. So it seems like the number of species is controlled by oxygen, at least in the Pacific, and the distribution of species is more controlled by temperature. When we went back and looked at carnivory questions, we found the same pattern, that oxygen was the best explanatory variable for the number of carnivorous taxa, but temperature was the best explanatory variable for the percent of carnivorous individuals. Again, a similar kind of result. And I'm going to close. I really don't have time to talk about other approaches for untangling these multiple stressors and looking at the importance of oxygen. I just wanted to mention that our lab has worked on these lab experiments that with mussels and squid that modify control temperature and modify CO2 and O2 together because they co-occur and change together off California. I'd be happy to talk to anybody about that. And another area that we've been working on is the development of geochemical proxies for exposure to low pH and low oxygen. And I'd be happy to talk to people about that work as well. Uranium to calcium looks like it could be a really good indicator for pH exposure, at least in bivalve shells. And the last thing I want to say is that I've been talking only about climate variables, but we know there are many other human stressors on continental margins and on upwelling margins: pollution, overfishing, certainly eutrophication, but species invasion and other forms of habitat degradation. And so I think we have to think more holistically about managing systems. And we have to build hypoxia into that management regime, but it has to be done in a multi-stressor context. Some of the things I think are some thoughts to leave you with are the importance of unifying the scientific expertise we have for hypoxia in the coastal ocean with the expertise for hypoxia in the open ocean. Those two communities have traditionally been very, very separate and not compared notes or studied together. But I think that I haven't talked a lot about the fact that warming is changing hypoxia in coastal and estuarine seas regions as well. So these influences of eutrophication and CO2-driven hypoxia are coming together. I think that we need to think about how to manage for this, and we can't control upwelling except for trying to reduce CO2 emissions, and we need to do that, and we can control nutrient inputs. So we know there are some things we can do about hypoxia. I think we need to figure out which species are sensitive and try to reduce the fishing pressure on those, and maybe refocus and maybe think about new climate-tolerant fishery species. I think there's an interest in using marine reserves to improve resilience, but people don't think about it in terms of hypoxia management, but I think they probably should. We need maybe temporal refugia when there's seasonal hypoxia, which there is even off Southern California and off the whole U.S. West Coast, as well as here. And the other thing is that we need to do more oxygen monitoring. It's amazing to find that there's such great biological data out there, and most of the time people don't measure oxygen along with the animal data. And I think that in that vein, I wanted to alert people to the fact that the Intergovernmental Oceanographic Commission, the IOC, is setting up a global ocean oxygen network with the goal of bringing together coastal and estuarine and open ocean researchers to try to raise awareness, combine data, maybe develop a meta database and form an observing network. I'd be happy to talk more about that to people if they're interested. And sorry, I've gone on quite a long time with that. I just want to thank you for listening, and I'd be happy to answer any questions.
Denitrifying either through symbionts or other, are they utilizing the nitrate that might be available?
That's a really good question. Probably the forams there are doing that, the foraminifera. I don't know. There's no evidence that those animals have symbionts. They eat lots of phytodetritus. But whether animals do other animals have the ability to denitrify?
There's some evidence out there now that some of them are harboring symbionts that are doing it.
That could do that.
Earthworms do it, I know, and oysters and other organisms.
So are you asking about the oligochaetes or the amphinomids, or any of them? Yeah, the denitrification maybe in the oligochaete ecosystem might actually, I'd have to go back and look at those papers to know whether they're doing that, but that's a possibility. I don't think anybody, I know nobody's looked in the amphinomids. Yeah. Yeah.
So I thought it was interesting that you saw that kind of expansion of trophic diversity in the oxygen minimum zones using stable isotopes. And I was wondering if you looked at sort of traditional functional guild, like how many suspension feeders versus deposit feeders. Is it that there's more sources of food, or are there more behaviors to get food?
Well, my interpretation would be that there are more sources with wider range of isotope signatures at the base of the food chain. I'm working with Eric to look a little bit at that, but we haven't done it yet. We know that there aren't carnivores there, and that what's really interesting is species that are traditionally carnivorous, like the amphinomids, shift to being phytodetritus feeders in these environments. I don't know whether it's an energetic issue or simply what's available. Like, they're the only species there, so they'd have to be eating each other to be carnivores, right? They're probably myofauna there to eat as well, but not many though. Yeah. So anyway, we're working on it. I could talk to you some more about it. Yeah.
Following up, do you think there's maybe a connection with body size and trophic roles? But what happens in shallow water, I think, is a lot of times because you have smaller body size being preferred, and they tend to also be the surface odd feeders. There can be sort of like a covariation in those.
But you're thinking smaller body sizes would expand trophic diversity?
No, I'm wondering if maybe with the trap too.
Well, that was our hypothesis, that we should see less trophic diversity, and we just don't see it with the stable isotopes. That doesn't mean that other functional approaches wouldn't see it, but it does look like there's a broader range of food types down there. Whether that might mean that if we just looked at the the diversity of primary consumers, we'd see expansion, but if we went only and looked at higher order consumers, maybe we wouldn't see that. So, yeah. Yes.
So since oxygen appears to be so critical in the evolution of many organisms, with the lack of oxygen or the disappearing of oxygen, is that going to be another contributing factor to sort of a massive extinction?
Yes, could, if it continues throughout the whole ocean. I mean, clearly things move away if they can, and go live somewhere else, right? But if there's nowhere else to go live, then you end up with tremendously reduced diversity. Now, even during our mass extinctions, we didn't mostly lose all life, right? We had some. If we use the core of the oxygen minimum zone as a model for what a mass extinction could look like, we know that there would be a handful of species still there. The fish I'm not so sure about. One of my students is working on oxygen minimum zone fish, and I used to think that we only saw fish there because they could swim out and repay their oxygen debt. But that's not true. It turns out that oxygen minimum zones have fish that can literally live at a couple of micromolar of oxygen probably their entire lives. So there are some amazing adaptations that fish have, but there's only, I think she's now found 50 species in oxygen minimum zones or something like that, 56 species. So, and at the lowest levels, there's about 5 fish species that can do that. So, yes, but we don't know. I have to say the modelers, I hope there's no serious climate modelers in this room. They're terrible at replicating the modern ocean oxygen conditions. We don't understand enough of the biogeochemistry and interplay with ocean physics and biology to actually predict what's going to accurately predict what the ocean looks like now, let alone what it's going to look like, at the end of the century. So we can't say yet whether for sure we're going to keep on losing oxygen, but we're going to do it for a while. Yeah. Yeah.
Do you think that at that oxygen minimum zone, we see that kind of staggering of maximum liver as you go up in body size? Some of that might be due to like predation at a northeast thing, so these things can tolerate low
Yes.
oxygen and maintain predators.
I think you're right. I think those species interactions are a big part of it. What you actually have to see it to understand that all this phytodetritus comes down, and when the water column's severely hypoxic, there are no zooplankton to use it up. So it just comes down, and you have phytodetritus piles on the bottom of the seafloor. And in that area, there's a tremendous amount of food. So if you have a lot of food and you have a species that's tolerant, they get to be really abundant as long as there's no predators, right? And so that size zonation, I think the absence of the larger predators at each level is really important to allowing that peak occurrence. Yeah. Let's see if there's anybody else. Nope. Go ahead.
So if there's that much phytodetritus, why is there chemosynthesis happening there? What's the advantage of doing that if there's a lot of food already there?
That's a really good question. And the chemos, you mean the symbiont-bearing chemosynthetic organisms?
Yeah, or just what leads to that spread in.
I think that somehow at really low oxygen levels, you need some oxygen for chemosynthesis that involves oxidation, right? Although some species can use nitrate, maybe some of those species can use nitrate instead of oxygen. But I don't know energetically what the pressure would be, but there might be some physiological explanation that we don't really quite know about yet. I mean, it's a really good question. They don't do it just for food. I mean, I've seen it off California. We've got chemosynthetic symbiont-bearing species living in our oxygen minimum. Some of those I showed living in our OMZ when there's plenty of other food to eat. They could filter feed. They can do other things. So maybe there's another benefit. Yeah. Yeah.
So here in the Gulf, we're kind of used to jubilee events with like kind of the fish and the crustaceans kind of coming out. That's like a big thing around here. But for other parts of the world, how prevalent is that? Like with the hypoxia drawing fish and crustaceans onto land and kind of like causing a stir?
It's pretty common in South Africa. Those lobster walkouts happen kind of regularly, I think, in the particular bays where they're subject to intense algal blooms. I've seen all sorts of stuff land on the beaches on La Jolla, where near where Scripps is. I've seen them covered with tuna crabs, red crabs. I've seen them covered with squid. Nobody's been measuring to know if they're escaping oxygen. That's not always the explanation, and in my mind, it often probably is, escaping low oxygen. But some of you may know better than I, how common on the Atlantic or other parts of the Gulf Coast it happens. Oregon started to see a little bit of that, and off Baja California, I've heard reports of it. Usually, it happens when there's this confluence of events that conspire to make the oxygen really low, like not just one thing, but all the weather conditions at the same time, the current conditions, the upwelling, the nutrient inputs all come together, and then, pow, you get anoxia and things die and wash up on shore. Yeah.
In the beginning, you showed the identification of oxygen minimum zones kind of through the decades, and you showed how a lot of them are centered around North America and Europe. How much of that do you think has to do with a lot of research being focused in those areas, and if so, how does that play out in our understanding or lack of understanding throughout the rest of the world?
So you're talking about the Diaz tracker. Those are not oxygen minimum zones. Those are eutrophication-induced coastal hypoxia. But it's a good question about whether that increase is really a temporal increase or more people started measuring oxygen and noticed it. And I would guess that there's some of both. I mean, we've had a lot of population increase since the 1960s, and probably, especially in less developed parts of the world, that there is probably some of that is just people making new measurements. But I don't know. You would almost have to ask Bob Diaz that question.
We have time for one more question.
I think we're good.
A number of questions. This is a good indicator of the quality of the talk. So, Dr. Levin, you must have given a good talk.
Okay, thank you.
Don't forget about the workshop today at 2:30.
Yep.
So you have more questions, please feel free, and we can always try to talk to
Yeah, let me just say one thing. I have to fold them, but if anybody was at my talk last night and is interested in having a DOSI brochure for the Deep Ocean Stewardship Initiative, I brought some with me today that I could give out. Yeah.